Every solid dosage form is engineered to deliver a concentrated chemical payload directly into the bloodstream. Because of this immediate physiological impact, every new batch begins its life cycle in a state of locked quarantine, physically isolated until its safety is documented. To clear this status, quality assurance subjects the material to a multi-stage analytical sequence, providing the evidence required to either release or destroy the batch. The process begins with starting materials, the active pharmaceutical ingredients and excipients. These powders must pass initial identity and purity checks before they are permitted to be formulated into a bulk product. Once approved, these powders are moved to production. A rotary press stamps the bulk formulation into individual tablets, relying on precisely calibrated mechanical pressure to ensure uniform density. Ejecting a formed pill from the press is only the initial step. To ensure the dosage form can survive high-speed packaging, transit, and eventually human digestion, its structural integrity must be mathematically verified. Quality control begins with friability. This test measures the tablet's ability to resist surface chipping and abrasion caused by friction during the packaging process. Technicians place a pre-weighed sample into a rotating drum, tumbling the tablets at 25 revolutions per minute for exactly 4 minutes. The compendial limit is strict. If the mass loss from broken pieces and dust exceeds 1.0% of the initial weight, the entire batch fails inspection. Next is the hardness or breaking force test. This measures the diametrical load required to physically crush the tablet, with an acceptable range typically falling between 5 and 10 kilograms per square centimeter. While the tablet must be durable for shipping, it must also break down quickly once ingested. Disintegration testing ensures the compressed core disperses into smaller particles in a fluid medium, the prerequisite for drug absorption. To simulate the human digestive environment, the units are submerged in a liquid medium maintained at a temperature of 37 plus or minus 2 degrees Celsius. This testing hardware is standardized by international pharmacopoeias, utilizing a stainless steel wire mesh basket and a solid paddle apparatus to create uniform movement. These schematics detailed the exact engineering required for the baskets. For the test to be valid, the wire mesh screen at the base must measure between 1.8 and 2.2 millimeters. Enteric-coated tablets follow a different protocol. When placed in simulated gastric fluid at a highly acidic pH of 1.2, they must show 0 signs of disintegration or softening for up to 2 hours. Passing these structural tests confirms that the tablet can reach the patient intact. However, evaluating the stability of liquid formulations requires a different set of analytical tools. Oral liquid systems, like syrups and suspensions, are complex physical dispersions that require constant monitoring to prevent chemical degradation or sedimentation. Their pH must remain within a narrow margin of plus or minus 0.05 units. Significant shifts in acidity can cause the active ingredients to degrade or precipitate out of the solution. Technicians monitor viscosity using glass viscometer tubes. This metric ensures the product pours easily and suspended particles don't sink too quickly during storage. Emulsions present a unique challenge. These are blends of immiscible liquids, oil and water, that are held together by an emulsifying agent. To identify the emulsion type, QA uses the dye partition test. Water-soluble methylene blue will spread through the background of an oil and water mixture, while in a water and oil emulsion, the dye remains trapped inside isolated droplets. If a syrup or emulsion fails these tests, the batch is rejected. But for products meant for injection, the requirements for safety are even more stringent. Intravenous drugs are produced in an engineered clean area. Personnel work in protective suits under sterile hoods, preventing any airborne particles from contaminating the product. The standard for sterile products is absolute. They must be demonstrably free of viable microorganisms, foreign particulates, and fever-inducing biological components. To verify glass ampule integrity, technicians use the leaker test. Ampules are submerged in dye under vacuum pressure to check for microscopic leaks. Beyond physical sterility, the product must be free of pyrogens. These are metabolic products of bacteria, endotoxins, that can trigger a fever response in patients, even if the bacteria themselves have been killed. Industry detects endotoxins via the LAL test, It uses horseshoe crab blue blood, which contains a lysate reacting to endotoxins even at picogram levels. Because these biological components are heat-resistant, depyrogenation of glassware requires a specific thermal cycle, baking at 180 degrees Celsius for at least 3 hours. The final validation is the sterility test. Samples are incubated in culture media for 14 days to provide documented proof that no microbial growth occurs. In the sterile zone, a single microscopic failure results in the immediate rejection of the batch and a mandatory facility-wide investigation. Some potent drugs are too complex for standard chemical analysis. The extract from the digitalis flower, for example, requires a biological check to determine its true strength. Manufacturers use a guinea pig bioassay to calibrate the dose. By measuring the biological response, they can compare the new batch against a standardized 76 mg reference powder. These assays allow manufacturers to quantify complex drug potencies that cannot be measured by chemical assays alone, ensuring a standardized dose reaches the patient. After all tests are complete, the batch documentation arrives at the QA desk for reconciliation. This process compares the actual yield against the theoretical yield to ensure no material was lost or mixed up. QA also verifies the content uniformity. This confirms that the active pharmaceutical ingredient in each individual dose falls between 85 and 115% of the label claim. With the evidence compiled, the batch reflects its required limits. Friability is under 1.0%. Hardness is within the 5 to 10 kilogram range, and the 14-day sterility test is clear. Once all documented standards are met, quality assurance lifts the quarantine. The batch is officially released, transforming a controlled chemical mixture into a life-saving medicine.